Well, let's look at all of the trials that are ongoing in hemophilia A gene therapy. I've already talked quite a bit about the BioMarin trial, and what I'd like to comment on is in these 5 trials that there's some similarities and a number of differences. 2 of the trials, the AAV, the recombinant AAV is made in insect cells, and in 3 of the trials it's made in mammalian cell lines. All of them are codon optimized in B domain deleted factor VIIIs. In one trial, a modified B domain deleted factor VIII is being used. It has additional glycosylation sites designed to increase the efficiency of processing of the factor VIII protein. With the idea that perhaps lower doses of vector might be used or higher amounts of factor VIII may be achieved. And so this is still in early clinical testing. Most of these have concluded their phase 1-2 trials. As I mentioned, the BioMarin study, the phase 3 is at its 1-year time point this past month. And then an additional phase 3 trial has gotten started with Sangamo Pfizer. Spark has indicated that they are doing additional phase 2 studies to try to sort out some of their data, which I'll discuss in a few moments. And then the additional trials are still in phase 1-2. Most of the patients have had elevated transaminases, not particularly high, but high enough to indicate that there may be an immune response ongoing that could be affecting the transduced cells with a loss of hepatocytes making factor VIII. Therefore, these patients were placed on steroids, and I'll show some of those data as well. And then with regard to durability, there is a lack of durability in some patients, and I'll show some durability in other patients. But throughout, we're looking at high intra-subject variability. And what you can't see here is also high intra-subject variability over time as well. And in many cases, the trials are small enough that we really don't see a clear-cut dose response due to that variability.